Chapter 4 Animal Kingdom. When you look around you, will observe different animals with different structure and form. As over a million species of animal have been described till now, the need for classification become all the more important. The classification also help in assigning a systematic position to newly described species. 4.1 Basis of classification. In spite of difference in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of column, pattern of digestive, circulatory or reproductive system. These features are used as the basis of animal classification and some of them are discussed here. 4.1.1 Level of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponge, the cells are arranged as loose cell aggregate. They exhibit cellular level of organization. Some division of labor occur among the cells. In coelenterators, the arrangement of cell is more complex. Here the cells performing the same function are arranging into tissue, hence is called tissue level of organization. A still higher level of organization is that organ level is exhibited by member of Platyhelminthes and other higher phyla where tissue are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, molluscs, Echinoderma and chordates, organs have associated to form functional system. Each system concerned with a specific physiological function. This pattern is called organ system. Level of organization. Organ system in a different group of animal exhibit various pattern of complexities. For example, the digestive system in Platyhelminthes has only a single opening to the outside of body that serve as both mouth and anus and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. Open type, in which the blood is pumped out of the heart and the cell and tissue are directly bathed in it, and second, closed type, in which the blood is circulated through a series of vessels of varying diameter, in bracket arteries, veins and capillaries. 4.1.2 Symmetry. Animal can be categorized on the basis of their symmetries. Sponges are mostly asymmetrical, is that any plane that passes through the center does not divide them into two equal wholes. When any plane passing through the central axis of the body divided the organism into two identical wholes, it is called radial symmetry. Coelenterators, Ctenophora and Echinoderma have these kind of body plan. Figure 4.1a. Animals like annelids, arthropods, etc., where the body can be divided into two identical left and right whole in only one plane, exhibit bilateral symmetry. Figure 4.1b. 4.1.3 Diploblastic and triploblastic organization. Animal in which the cells are arranged in two embryonic layers. An external ectoderm and internal endoderm are called diploblastic animal. Example, coelenterators and undifferentiated layer mesoglea is present in between the ectoderm and the endoderm. Figure 4.2a. Those animals in which the developing embryo has a third germinate layer, mesoderm, in between the ectoderm and endoderm, are called triploblastic animal. Platyhelminthes 2 quadrate, figure 4.2b. 4.1.4 Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animal possessing coelom are called coelomates. Example: annelids, mollusca, arthropods, echinoderma, hemichordates and chordates. Figure 4.3a. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouch in between the ectoderm and the endoderm. Such a body cavity is called pseudocoelom, and animals possessing them are called pseudocoelomates. Example: Asclepianthus, figure 4.3b. The animal in which the body cavity is absent are called ocoelomates. Example: Platyhelminthes, figure 4.3c. 4.1.5 Segmentation. In some animals, the body is externally and internally divided into two segments with a serial repetition of least some organ. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomena is known as metamerism. 4.1.6 Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development. In some animals with notochord are called chordates, and those animals which do not form this structure are called non-chordate. Example: Porifera to echinoderms. 4.2 Classification of animals. The broad classification of animalia based on a common fundamental feature as mentioned in a preceding section is given in a figure 4.4. Kingdom Animalia in bracket multicellular. Level of organization: cellular level, tissue, organ system. Level of organization: cellular level. Symmetry: mostly asymmetrical. Body cavity or coelom or coelomata. Phylum Porifera. Animal multicellular. Tissue, organ system. Radial symmetry, bilateral symmetry. Radial symmetry: coelomata, coelenterata, ctenophora. Bilateral symmetry: without body cavity, coelomates. Platyhelminthes with false coelom, pseudocoelomatus. Asclepianthus with true coelom, coelomates. Annelida, arthropoda, mollusca, echinodermata, hemichordata, chordata. Echinodermata exhibit radial or bilateral symmetry depending on the stage. Figure 4.4 Broad classification of kingdom Animalia based on the common fundamental feature. The important characteristic feature of the different phyla are described. Phylum Porifera. Members of this phylum are commonly known as sponge. They are generally marine and mostly asymmetrical animals. These are primitive multicellular animals and have cellular level of organization. Sponge have a water transport or canal system. Water enters through the minute pore in bracket ostia in the body wall into the central cavity spongocoel form, where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Coenocytes or collar cell line the sponge coil, and the canal digestion is intracellular. The body is supported by skeletal made up of spicules or spongine fibers. Sexes are not separated. Hermaphrodite is that example, and sperms are produced by the same individual. Sponge reproduce asexually by fragmentation and sexually by formation of gamete. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult.